Welcome to the podcast of HemaSphere, the official journal of the European Hematology Association. HemaSphere's podcast presents insightful, expert discussions about recent hematology publications. We hope you enjoy.
Hi, and welcome to the HemaSphere podcast. I'm your host, Charles de Bock. When it comes to understanding leukemogenesis or hematopoiesis, mouse models have been a critical part in terms of being able to overexpress genes or knock out genes and then look at the downstream functional impact of those on either blood cell development or the development of leukemia. For example, you might want to overexpress a certain oncogene and clone that in the ROSA26 locus, which is a safe harbor, and then express that constitutively or inducibly using a lock-stop-locks cassette, and then make use of the many lines that are available to express that gene within your favorite blood lineage. Of course, we can also use CRISPR to knock out putative tumor suppressor genes and then follow whether that accelerates leukemia or again perturbs blood cell development. Right next to that, the advent of immune-compromised mice like NSG mice or NOD/SCID gamma mice have been critical in our ability to engraft human leukemia cells and then generate what's commonly referred to as patient-derived xenografts. And so why is this important? Well, these have been really able to advance our knowledge on clonal heterogeneity and tumor biology, but also their utility in testing preclinical drugs or drugs that you want to test to see if they can decrease leukemia. And so these PDXs or avatars have been really important in our ability to assess the efficacy of new targeted agents against either standard of care chemotherapy or other combinations of drugs. And so it's with great pleasure I am joined today by Professor Richard Lock, who is from the Children's Cancer Institute in Australia. And he has been part of the US NCI-funded Pediatric Preclinical Testing Consortia for about 20 years and is a world leader in using PDX models and immune-compromised mice and in preclinical testing. So welcome, Richard, and thank you for joining us today.
Thank you, Charlie. Great to be here.
So we have a very broad audience, and some of the topics I would like to cover today is, your experience over 20 years on preclinical testing, the limits and utility of PDXs, and so we can ask those questions about how useful they are, but also understanding, what they're not great for, and then hopefully ending on some of the other topics, which is what about AMLs and more rare cancers, and also the use of testing immune therapies, which is obviously really taking off currently in the clinic. So perhaps to start off with, I'm going to ask you just to outline what the NCI-funded Pediatric Preclinical Testing Consortia is and how you got involved and why it got started.
Yeah, thanks, Charlie. So about 20 years ago, it was recognized that there are effective therapies available for many childhood malignancies, but there are certain high-risk subtypes that really don't do very well at all. And there are many new agents in the adult cancer setting that are available for testing against childhood cancers. But, childhood cancers are relatively rare, so there simply aren't enough pediatric cancer patients to test all of these potential active agents in clinical trials in kids, and certainly that's not ethical. And so the NCI-funded preclinical testing consortia came about in an effort to generate high-quality preclinical testing data that could be used to inform clinical decision-making as to which agents should be fast-tracked into clinical trials in children with cancer, and certainly which agents shouldn't be tested in children if they show no activity in preclinical models. This consortia was designed to be primarily an in vivo testing consortia. It's not a basic science consortia. It's actually, a preclinical testing consortium. And so, as I've said, in regard to prioritizing new agents to go into the clinic, the consortium realizes that it's important to publish data on inactive agents, because if a potentially inactive agent is moved into a clinical trial, then it may delay testing of a potentially beneficial drug. And more recently, the RACE Act, which is the Research to Accelerate Cures and Equity Act in the US, now mandates pediatric cancer testing. So about 20 years ago, the NCI made available considerable funds for consortia to bid for funding to undertake a preclinical testing program. We were originally called the Pediatric Preclinical Testing Program, and we had 2 rounds of 5-year funding. Then it was called the PPTC, which is the consortium, and in its current iteration, it is called PIVOT. So that's another acronym for Pediatric In Vivo Testing Consortium. So our patient-derived xenografts, or PDX models, recapitulate the gene expression profiles and genetic alterations common to childhood cancer and supporting their value in drug development.
Amazing. So, now I think you've got at least 90 that you regularly use, and that's expanding to over 135, I think, at last count, but building on that all the time. And we're going to get to that in terms of how to start off if you're new to the PDX field and you want to make your own bank, or you've got a bank and want to understand how to maximize that. But perhaps for those that don't fully understand how a PDX is made or how you start, perhaps I can ask you just to give a very brief outline of when you first started, how you began, and what are some of the key things to generate your very first PDX? What do you actually need to do when you make a PDX?
Well, you need to collaborate with good people, and that's essential to do good science. But it's a very straightforward process. It's as the name suggests, it's patient-derived, so it's patient-derived material, whether it's a solid tumor or a liquid tumor like a leukemia. Very rarely are these cells cultured ex vivo before they are inoculated into mice. It's essential to have the correct strain of immune-deficient mouse. And so at the time, we had the opportunity to collaborate with researchers who were interested in engrafting cord blood into immune-deficient strains of mice. We therefore collaborated with these researchers and decided on a program of trying to establish PDXs from acute lymphoblastic leukemia, principally bone marrow aspirates, but occasionally peripheral blood will engraft in mice. And in terms of leukemia models, over the years, they've been recognized as the gold standard for preclinical testing, because when you inoculate these cells into the tail vein of an immune-deficient mouse, such as an NSG mouse, they home to the bone marrow, and then they take over the bone marrow and they disseminate to the spleen and other organs such as the liver, and then they disseminate to the peripheral blood. Now, a lot of people also use cell line-derived xenografts, and there are many instances and reports in the literature of commonly used cell lines that are inoculated subcutaneously, and so you get a nodule of leukemia. But in our PDXs, we get a systemic model of disease. So essentially, the mouse develops a human leukemia. The PDXs retain the cell and molecular characteristics of the original disease. You can monitor engraftment and drug responses in real time by taking tail vein bleeds and conducting flow cytometry, because the levels in the peripheral blood give an overall good representation of the leukemic burden in the animal. And there are some PDXs that we've transduced to express luciferase, so we can carry out bioluminescence imaging. And really, over the years, we've developed the ability to create PDXs from specific patient subgroups, so we can generate 1 PDX per patient. And as you say, we've now developed more than 150 PDXs, and we currently have 130 that have been molecular annotated.
So that's the key, right? In terms of, in my mind, the molecular characterization, and that it still recapitulates the primary disease in terms of what was in the patient. You don't really get clonal selection where a very rare clone, well, perhaps you can comment, how often do you get, if you look at the genetic structure of the primary patient at diagnosis and then in the PDX, do you see significant genetic drift when you establish the PDX? How well does the PDX actually marry up to the primary patient at diagnosis? Do you see much in change?
Yeah, overall, these PDXs do reflect the primary disease very faithfully, but I think we have to recognize that there will be some selection in the mice. And I think it would probably be naive to think that there wouldn't be some sort of genetic variation in the PDXs. And that's why it's critical for one to understand the molecular annotation of the PDXs. If you are going to design preclinical testing, you have to know what you're working with. And in some of our earlier studies, we actually looked at PDXs that were derived from patients who subsequently relapsed. And what we found was that the PDX in the mouse evolved to be more like the relapse clone. So what it said to us was that the PDXs in the mice were actually behaving as per the outcome of the patient. So because, as we know, not about 97% of pediatric ALL patients respond completely to their initial treatment, but then some of them undergo relapse. But what we found in those PDXs from patients who subsequently relapse were that they developed into aggressive PDXs. They were chemo resistant. So I don't believe that if you take a diagnosis sample and you inoculate it into an NSG mouse, it is going to stay exactly the same diagnostic sample.
Yep. And what about when you serially passage them through secondary and tertiary transplants? Are they, after the primary one, do they generally stay quite genetically stable thereafter, or do you keep getting continual selection? That might be reflected by the time to engraftment, or do you see drift over time?
Yeah, in the limited number of studies we've done, they actually do stay remarkably stable. And what is quite fascinating to me is that every PDX has its own rate of engraftment, in that after inoculation, there's a lag phase before you start seeing leukemia cells appear in the mouse peripheral blood. And that is a very stable characteristic of each PDX. So, for example, several years ago, we had a PDX derived from a patient at relapse that took about 110 days to appear in the peripheral blood. And we reasoned that if we keep doing serial engraftments, one day that PDX is going to transform into a rapidly engrafting and very aggressive PDX. And I think we went up to about 10 serial passages in the mice, and it still took 110 days to appear in the peripheral blood. And if you multiply 10 passages by 110 days, that took one person a long time to do. And in the end, we gave up in trying to get it to transform. So yes, after you get through the initial engraftment, they do stay remarkably stable.
Which is probably a good thing, especially if you've got like a bank of them, so you can have, I guess, confidence in your results, even if you're using a bank of tertiary or quaternary transplants.
So in our processes, we've realized that when you do the primary engraftment, which is to develop the primer graft, that's actually a limited resource, because if you utilize all of that primer graft, then you have to go back to the original patient sample, which very often isn't available. So in our standard procedures, we would always take that primer graft and then expand it into secondary and then tertiary recipient mice for use in our preclinical drug testing. And those are the PDXs that we've had all of the molecular annotation done on, and that all of those data are in the public domain if anyone wants to interrogate those PDXs.
So when you talk about the molecular annotations of your primer grafts, and then obviously you can use that to quality control for your secondary and tertiary, because inevitably you want to make sure if you find an anomaly, you can track back again why it's so important to kind of maintain that primer graft. And I guess that, if you're listening and you're making your PDX for the first time and you're doing the initial PDX generation, is protect that and then only use secondary and tertiary transplants for your working stocks and your following up experiments. So when you're talking about molecular annotations, what do you actually mean in terms of what extent have you characterized molecularly your PDXs to date?
Well, one of the fundamental questions that we wanted to ask on a large number of PDXs was, do the molecular lesions in those PDXs reflect the general population of what we would see in pediatric ALL? So do they have the common fusions? Do they have the common mutations that are associated with relapse, etc.? So for all of these PDXs, we've had copy number variations, we've had structural variants, we've got single nucleotide variations, and we've got RNA-seq for mRNA expression. And so, as I've said, that's all available in the public domain. So if you wanted to test a molecularly targeted agent that targets a particular gene fusion or a particular mutant protein, you can search for that gene on the publicly available site and identify the PDX. And then we make our PDXs available to the academic community if they wish to do their own preclinical testing.
Yep. And I think it's on our PedcBioPortal, I think is the place. So if people are wondering where you find it, it's PedcBioPortal. You only need a Google account, I think, to access that data. There's a lot of data there, and I think if you scroll down, you'll find all the leukemia samples, and there's 90 that are in that public domain.
And there are also sites such as PDX Finder. There's a huge resource out there. There's a lot of people now making leukemia PDXs, not just us. So it's
excellent. And it's really great that you're willing to share them, because I think that's fantastic for the patients as well, to find out that, for their rare cancer, there might be a group that's working on that in terms of trying to find a new therapy. So if I go back to the preclinical testing part, which is now becoming more and more common within a lot of publications as people establish PDXs and then test their favorite agent to generate a Kaplan-Meier survival curve. And as part of your testing procedure, you have what's called objective response criteria, which is, I find, really, really fundamental. And like yourself, I review papers, and I think it's setting that bar in terms of understanding before you test what you're actually looking for in terms of how you define event when you start treatment. So can you just explain, for those who are not familiar, what the objective response criteria are, and when you would probably want to use it and when you maybe don't want to use it if you are using perhaps a tool compound per se?
Yeah, so very early in our consortium, it had been recognized that there had been a lot of failures in terms of mouse models over-predicting responses to novel agents. And if you think about it, if you have the luxury of a vehicle control treated cohort and then a drug treated cohort of mice, you don't have that luxury in the clinic based on one patient. And if you are just looking at progression delay of that particular leukemia, if you think about it in the clinic, that's going to be progressive disease for the patient. So it was really important to us as a consortium for both the solid tumor and the leukemia models to actually be measuring regressions. And these are the objective response criteria that were modeled after the clinical setting. So simplistically, an objective response is coded as an actual regression of the solid tumor down to less than 25% of the initial starting volume. And in the leukemias, it's a regression of the leukemia in the peripheral blood. So we've coded these objective response criteria in terms of partial response, complete response, and maintained complete response, rather than just trying to measure significant progression delays that may be statistically significant, but they're probably unlikely to be biologically significant. And it's a very high bar to get new agents into clinical trials in children, especially ALL, because there's many active agents in the pediatric ALL field. And so we only want to be accelerating the most potentially active new agents into clinical trials in kids with ALL.
And I think, one of the things that are highlighted in your most recent publications were the menin inhibitors that you helped test, which are now in November were approved by the FDA. So that's revumenib, which you and Scott Armstrong, were critical in showing that was really, really efficacious as a single agent across a panel of PDXs. And so on that, one of the things in that particular paper, because people do conventional testing, which is they might take 5 or 6 mice and then test their agent and then look for a survival benefit. And then, as you mentioned, progression delay or statistical significance may or may not be important to the question. And they don't monitor, in terms of regression. So you start at 1% in the blood. At that stage, the bone marrow is probably fully engrafted above 50 or 60%, and there's engraftment in the spleen, and that's the measure. Other people start at a different rate, which is fine. But I think one of the amazing things that you've pioneered in part is the single mouse trial, which is, rather than using 1 PDX and 5 mice, is that you have 1 PDX and 2 mice. One is the one gets the targeted agent, and one is the vehicle. And then you have a panel of PDXs, which is almost like a real clinical trial where each PDX is a specific subtype. So how did you come up with the single mouse trial, and how has it helped revolutionize, I guess, the testing in your eyes over and above the conventional testing?
It's just been a fantastic development, but I would say to anybody embarking on this venture now that you really have to have well-characterized models, because you don't want to be building castles in the sand, because the ALL PDX models are highly reproducible. And when we've done retrospective analysis of taking random mice from hundreds of previous experiments and comparing that random mouse response to the actual median of the group that it was included in, we get a 90% concordance between the single mouse and the median of the group, meaning, and what that means is we can do these single mouse trials with 90% confidence that the response of that mouse would have been reflective if we had done a group of 6 mice. So instead of now carrying out a conventional testing of 6 mice, control and treated, on perhaps 4 PDXs, we now routinely carry out single mouse trials where we have 1 control vehicle mouse and 1 drug-treated mouse per PDX, and we're routinely now testing around 30 PDXs, which gives us greater scope for what, it's almost doing preclinical testing on a clinical trial scale. And we've also done single mouse trials on 75 to 90 PDXs to generate preclinical data. And we have worked with a lot of companies over the years, and they are becoming very acceptable to this single mouse trial model. But I do have to have confidence that you are working with highly reproducible models. Just to illustrate with the ALL PDXs, you can very often get progression delays of, say, 5 days of the drug compared to vehicle control. And because the groups are so tight, you would get a significant p-value. But we all know that is not a biological significance. But if you have highly variable PDX engraftment, then you can't really do the single mouse trials in confidence. So you have to do your power calculations. You have to understand your models. And you have to set your event criteria in advance of this type of testing.
Yes, I think that's critical, especially setting your criteria for event prior. So that is either 25% CD45 in the blood, of human CD45 in the blood, or another criteria, but having that already set to allow you to have like an unbiased assessment of when, how efficacious your drug is in particular. So you're talking about the molecular characterization and this of ALLs. Now, not everyone is in ALL. There are obviously other rarer cancers, and I know that AMLs have been traditionally very poor engrafters in making PDXs. I think only about 30% have the ability to engraft, and most of those, of course, the aggressive and forms of AML, and so you don't get a wide representation of the different types of AMLs in particular, or even JMMLs or rarer cancers. So in terms of, the field as it is now, what have been some of the challenges to generate AML PDXs? And I know you've got an interest as well in making AML PDXs, and so how have you tackled making a better AML PDX panel?
We originally started with non-skid mice with the ALL PDXs, and then we moved on to NSGs, mainly for the reason that non-skids spontaneously develop lymphomas and only have about an 8.5 month lifespan. But NSG and non-skids are very skewed towards engraftment of lymphoid lineages. And historically, NSG mice have only had about a 20 to 40% take rate of AML, either adult or pediatric AML, compared to 80 to 90% engraftment rates for ALL. So it's obvious that we need to use improved mouse strains if we want to develop AML PDXs, and there are, there's a number of immune-deficient mouse strains that are now transgenic or knock-in for human cytokines that will facilitate myeloid cell engraftment. So now we're getting much greater success rates in engraftment of pediatric and adult AML primary samples, and even some JMML samples as well into PDXs, whereas previously, as I've mentioned, the low engraftment rates for AML, but also it was skewed more towards the aggressive and relapse samples, whereas now we get a very good success rate in creating PDXs from diagnosis biopsies from adult pediatric AML.
So to go back to those, once you've established them, and I just want to touch base on the actual testing, because, you've done a lot of different drugs. Some blockbusters include the menin inhibitors in your time, which is amazing. But also, you talked about, the one thing is that we have a lot of DNA damaging agents, and NOD/SCID and NSG mice aren't great for that, especially if you're going to make an AML and you want to use DNA damaging agents. What are some of the limitations of PDXs when you're testing drugs? What should you be thinking about in terms of the actual model itself, like not so leukemia, but the mouse, and how has that changed when you were starting off your preclinical testing and you realize, well, we can't use that drug because that's just wiping out the mouse because it's super sensitive. So what's been your experience, and what people should be thinking about when they're testing a new agent?
Yeah, well, the SCID mutation is difficult to work with if you want to use anthracyclines. So there's a lesson to be learned there. If you have access to RAG1 or RAG2 mutant strains of mice, you may have greater success in being able to use standard of care therapies for AML. We've learned over the years that PDX models can overpredict the activity of mitotic kinase inhibitors. We had a hard lesson when we used the Aurora kinase A inhibitor, alisertib, many years ago, and we got astounding results, and it was fast-tracked into a clinical trial in ALL and neuroblastoma, and it failed because we then retrospectively found out that the kids couldn't tolerate the doses that adults could, and our dosing in the mice was based on the adult clinical trial. So hindsight is 2020. So be aware of, you might get overprediction of mitotic kinase inhibitors because the PDXs will probably grow a bit faster than the tumor in the human. So it makes sense that you would get overprediction of mitotic kinase inhibitors. I would say you've got to be really cognizant of mice tolerating higher doses of many different drugs than humans will. So you cannot just do your preclinical testing at the maximum tolerated dose in mice. You have to consider murine pharmacology versus human, bioavailability of the drugs and plasma protein binding. If you want to have successful translation of preclinical results to the clinic, because you just don't want to be over-predicting the activity of a whole host of drugs that then go on to fail in the clinic. You're also working with immune-deficient mouse strains, so you don't have the immune component that you would in a genetically engineered mouse model or a human. You've also got a mouse microenvironment, so you have to be aware of that. And you may get some genetic drifts. So it's an imperfect system, but at this point, it's recognized as the gold standard. But I think if anyone is embarking on this now, you need to go in with eyes wide open and recognize the limitations of the models that you're working with.
Yep. And I think, one of the things as well is that, we've got a lot of immune therapies coming through, of which, as you said, in an immune compromised mouse, you can't test a bite, for example. You'd have to first humanize the mouse. And I know, for example, ADCs are coming in, and your group has a paper and press with HemaSphere, which is, congratulations, it's fantastic news. And that also used a single mouse trial. So if you're listening and you want to look at a single mouse trial in action, looking at an ADC, and that's using a CD123 targeted antibody for leukemia. But what about like CAR Ts and BiTEs? Like how do you move into that field, and what do you think the future holds for our ability to test those types of therapies, which are obviously becoming more and more popular?
Yeah, and just going back to the paper that is in press in HemaSphere, we did actually, one of the supplementary figures is comparing the single mouse trial data with conventional testing we did on the same PDXs, and those experiments were done at least a year apart, and the correlation between the single mouse and what the group of 6 mice responded to is quite startling. So, that reinforces to be able to use single mouse trials. But in terms of immunotherapy, there are mouse strains that you can engraft human T cells. So, we are moving towards being able to test these BiTEs by expanding human T cells and inoculating them just before treatment with a BiTE. One of the issues that you will face if you use NSG mice is that you are highly likely to get graft versus host disease and all kinds of sickness in the mice that may then have to be euthanized before you've been able to measure any kind of response. And so, go onto the Jackson Labs website and see the tools that they have available to now study oncology therapies in different strains of mice. For example, there is an NSG MHC1/2 double knockout that doesn't suffer from GVHD at anything like the rates that NSG mice do. So, that's the kind of strain that you would like to place your bets on if you're going to look at a BiTE or a CAR T cell rather than an NSG mouse.
Richard, this has been amazing. It is amazing insight. The future, where do you see preclinical testing for ALL in the next decade? What can you predict? And, as your final take on the utility of generating PDXs and how they've advanced our knowledge in the field of novel drugs. So where do you see the field heading in the next 10 years?
Yeah, I mean, there's still many new classes of drugs that are coming through. I was just at the ASH meeting and there were so many sessions on menin inhibitors. So that really is an exciting field that we played a lead role in. But we've got many new classes of drugs that are coming through to be tested in our preclinical models, degraders, molecular glues, etc. Targeted agents that is going to facilitate precision medicine. And I think, as I said, that you need to understand the molecular annotation of your PDXs, and I always recommend that you authenticate your PDXs either by SNP or STR so you know what you're working with. But then you can test targeted agents that may not be active against 90% of ALL cases, but if you can help one child with a particular genetic lesion survive their disease with a targeted agent, then I think it's still worth carrying out this preclinical testing. One thing that we haven't really discussed is proteomics and proteomics. At this point, we molecularly annotate the PDXs as I've described, but we really need, it's the protein where the business is done. So, we're now embarking on a project to generate proteomic profiles of all of our PDXs and then look at how the PDXs respond with respect to different drugs and the proteomics. So that's another huge area that we haven't really touched on.
I think you're right. We use RNA sequencing and genetics just so confidently in predicting as a biomarker, but in the end, drugs actually bind and target proteins, especially kinases inhibitors. They have to be active, and as many of our listeners will know, is that the expression of a particular kinase and its activity don't always correlate. And so you have to be very careful in using the expression at the RNA level and whether that's a good predictor of whether they'll be sensitive to a particular targeted agent.
Absolutely.
And so I totally agree that I think proteomic characterization needs to occur. And, it's obviously a bit more challenging because whereas RNA sequencing and genomic sequencing, you can readily access that with third-party providers, proteomics is probably just growing in that field where you can access that. And then, of course, the depth and the batch effects will also have parts. So I think that's indeed a fantastic way to move forward in understanding why a particular drug may work in one setting and not another one. Richard, thank you so much for your time. It's been a fantastic discussion on PDXs and the utility in preclinical testing. I hope listeners got a lot out of it in terms of their own research, and we would welcome you back maybe in a few years' time, Richard, to see if some of your predictions have come true. But otherwise, thank you so much for your time, and thank you to everyone who's listening, and join us next time for the next HemaSphere podcast.
Thank you for listening to this episode of the HemaSphere podcast. All of HemaSphere's content is open access and can be found at www.hemaspherejournal.com. We hope you will join us for future podcast episodes.